Now I would like to welcome our next speaker from Google, Michał Januszewski. Michał is a staff research scientist at Google Research in Zurich, where he is currently a member of the connectomics group. He's working on automating methods for high-throughput brain mapping at synaptic resolution. Prior to Google, Michał did research also in the fields of computational fluid dynamics and stochastical dynamical systems. Welcome, Michał.
Thanks. Thank you. Thanks so much for the kind introduction. And yeah, let's get started. So as was already mentioned, I'm a member of the connectomics team at Google, where what we are trying to do is to use Google's expertise and resources to advance our understanding of biological brains. And the way we are trying to do this is through acceleration of very high resolution brain mapping of specific examples of brains. So since we are also wanting to operate at a synaptic resolution, that implies the use of electron microscopes, because the features that we are trying to see are on the order of basically tens of nanometers or smaller than that. So our work is basically the development of algorithms to try to better understand those data sets, and also software to both store and visualize them. And I will tell you a little bit more about this a little bit later. We are a group that is a mix of research scientists and engineers, and also one student researcher. And one thing that is very specific to our group is that we collaborate very closely with academic partners, some of which you can see here on the slide. What we are trying to do is quite complex in order to go from a biological sample of brain to something that you have in a computer and that you can analyze. It's a multi-step pipeline. Basically, what you have to do is you, let me see if I can use a pointer here. Oops. Yeah. So basically, you start from a biological sample of brain that you have to extract, then you have to embed it in resin, you have to cut it into very ultra-thin sections, which you then have to put on a wafer, you have to image it into an electron microscope, and then there is this data processing that starts. You get raw images from the microscope, but you also have to stitch and align them into a uniform 3D data set, which you then have to segment, you have to visualize it, maybe you have to correct some mistakes that the automated methods have made, and in the end, you end up with a data set that you can analyze. So it's a multi-stage process, and usually the way we organize this work is that there's this first stage, which is done by our scientific collaborators, our academic collaborators, and then once the data analysis starts, this is where we come in and we try to help with those kind of challenges that are related to processing the data on a computer somewhere. All right, so just to be a little bit more specific and give you a little bit of a flavor of what we are trying to do. Oops. Did I? Perfect. Thank you. All right, I'll maybe not touch this to make sure it doesn't break again. Anyway, so just to give you a little bit of an example of what we are trying to do, this is the data that we're working with as it comes from a microscope after some light post-processing, so after stitching and contrast normalization. This is an image of an ultra-thin section of brain tissue. What you're seeing are cross-sections of brain cells, so of neurons and glia and other things that are in the brain. And so where were we? I think I was trying to tell you what we are trying to do with the data processing. So yeah, this is an image of a slice of brain tissue. And one of the problems that we are trying to solve is to figure out what the slice actually represents. And one of the way to do that is to segment the data. So what I'm showing you is an instant segmentation where every individual neuron has been assigned some random color. The color is meaningless. The differences between colors between different things are meaningful. And of course, this is just a slice through 2D data from a 3D data set. So really, if you're doing your job correctly, if you then take one of those things and visualize it in 3D, you should see a brain cell, so a neuron like this. And this is actually cut just because the data set is not large enough to contain the whole neuron. A neuron alone is not enough. What we also need to do is synapse detection, so also the points where different neurons connect to each other. There's quite a lot of them on every neuron, and once we have that, we repeat it over the whole data set, and we get a network, which is what we are really after. So the so-called connectome, that is a graph representation of connectivity between brain cells. And this is actually just a small fraction of the neurons that are contained within the data set, less than 1%. All right. So one question that one might ask here is, why do this in a collaboration, right? And in our experience, I can tell you that those collaborations, they tend to work best when there is a common goal that we are trying to solve, which in our case is easy. We are all trying to understand the brain better. They work well when the project is large and complex, which is also in our case somewhat guaranteed by the complexity of the processing pipeline. And third and fourth, which are also important points, there has to be complementary expertise in between the collaborators. So in our case, usually what works best is that we are the kind of data processing side, and our collaborators are the people who are experts in biology and in the data preparation, the physical data preparation, taking the samples. There has to be also some shared understanding between all those groups. So, I mean, we as computer scientists, we are not experts in the biology, and of course the biologists are not computer scientists, and they don't necessarily need to appreciate the difference between a transformer and a convnet, but we need to have a high-level understanding of what is a neural network, how do you use it, but you need ground truth data to train it, and but it doesn't produce perfect outputs, it has errors and so on. And similarly, we need to understand a little bit about this data preparation stages to know what sort of problems we can expect. Sometimes maybe sections are missing, or maybe artifacts from scanning or cutting or something. So we have to be prepared for that. And I don't need to understand the exact chemical processes that are used in fixation and staining, but I need to have a high-level overview of what's going on there. So if all of those things are in place, then the collaboration tends to work quite well. To give you some specific examples of collaborations that we have done in the recent past and the results, one of our largest projects so far, actually the largest project so far, is this human neocortex sample, just a cubic millimeter worth of tissue, which is tiny in physical space, but it's actually 1.4 petabytes worth of images that had to be processed for this, and the imaging itself took almost a year, and that was with one of the fastest electron microscopes in the world that was in existence at the time. The brain sample and the reconstructions that we have done with the Lichtman lab at Harvard, it's actually published now, so if you open this link in your browser, you can interactively see it on your laptop, and if you ever wondered what a human brain looks from up close, this is a very nice way to do that. One nice thing about this approach of, one nice outcome of this approach of doing things is that what we are getting is really a very comprehensive picture of what the brains contain. So we get really everything that is in there. We get all the neural cells, or the glial cells, the blood vessels, literally everything, which is very, very good for various types of analysis. And what I wanted to show you is some very curious things that we have found that nobody really expected before we started looking at the data. Some of them we still don't fully understand. So first of them is those kind of rare but more statistically frequent than one would expect, so-called multisynaptic contacts. So what you're seeing here is a particular spatial location where a given pair of neurons are connecting through synapses between an axon and a dendrite, and you can see that there are those multiple connections which are very close in space, and in some, you don't need to be a neuroscientist to appreciate probably that those things really are trying to make sure that they are going to communicate, which actually makes sense if you think about it a little bit. The synapses biologically are not very reliable devices, so if you want to make sure that a signal is going to pass from the presynaptic neuron to the postsynaptic one, it's actually good to have multiple independent connections, and here they're being made in a particular location in space. Another very curious thing that we have found that nobody really knows what it is are those axons that have this very kind of whirly convoluted shape, and we don't really know if this is a bug or a feature, so to speak, to use some computer science language. So it could be a sign of a disease, or it could be something that is functionally important. Nobody knows. As far as I know, nobody has ever seen this before, which is why it's unknown. But it's interesting. Nobody expected to see it in this data set. And then third thing that I found to be a little bit sad. So I always thought that cell bodies are topologically spheres. This is a counterexample. This is a dendrite that is literally piercing the cell body. So much for sphere topology. Biology is weird, and in a large enough data set, you're going to find a counterexample to almost every rule. So yeah, some interesting observations from this data set. And then another data set that I wanted to mention, this is a collaboration with HHMI Janelia, specifically with the FlyEM group. It's a reconstruction of a significant part of Drosophila brain. So this is a fruit fly. This is kind of the extent of the brain that was mapped compared to the size of the whole thing, maybe between 1/2 and 2/3. This is also publicly available, so you can, you can similarly to the previous data set, you can see it interactively in your browser. One thing that is different from, and I'm just going to let the animation play so you can see how complex the fruit fly brain is, even though a fruit fly doesn't seem to be a very complex organism. Maybe one thing that is different about this collaboration in comparison to this H1 data set is that this actually has undergone quite a lot of human proofreading. So there have been human experts on the side of our collaborators who have tried to correct as many mistakes as possible from our automated processing. It actually took about 50 years worth of work to fix all of the errors to the level that has been released in this data set. So yeah, fruit flies are tricky because the morphology of the neurons is actually more complex than that of the higher organisms like humans or mice and so on. So that also implies that the tracking of those neurons is harder. All right, so now to get to the machine learning part of things, I mentioned before that one thing that we are trying to do is to segment the neurons out. And this is one of those funny things that already 50 years ago, people were predicting that it would be solved in a couple of years. So this is from 1979, and people were claiming that it will soon all be automated, and humans will only deal with the hard cases. Now, in reality, in 2015, so some 40 years later, the state of the art was this on the left-hand side, right? So this is a single neuron. In an ideal case, it should all be one color and not have this blue thing here on the side and so on. So it didn't work that great. It's actually a lot of work to complete one of those neurons then manually through corrections. So we thought, okay, well, maybe, and this was already using machine learning, by the way. This is not like some, old school computer vision. This was actually state-of-the-art neural nets at the time. So we thought, okay, maybe there's a way to try to improve this. And indeed, if you put in if you put in a little bit of work, it's possible to get to at least this state, which is much better. You can see only a few colors, so with a few manual corrections, you can get a complete neuron in this case. And the contributions that our group has made to this field at the time is this thing called flood filling networks. So this is a relatively simple idea where you take a neural network, could be really anything. In practice, it's mostly a convnet, which is this thing here in the middle. And the trick is that you make the network take two inputs, which is the image itself and a so-called object mask. And then you ask the network to extend this mask spatially somewhere. And then you iterate this process until it terminates, so you run out of locations to fill. And this is a 2D visualization of what's going on. So the network is trying to kind of color in within the boundaries of a single neuron. In reality, again, this is a 3D data set that we're working in, so it actually works in 3D. This is a real recording of one of the kind of inference zones that we did on one of our data sets. And yeah, if it doesn't make any mistakes, you see it kind of tracing out a neuron or a neuron fragment in 3D. So we try to be quantitative about things, not just look at pictures, but also maybe numbers every now and then. And one way to quantify progress in this field is to measure how far you can trace those neurons before you make a mistake of some sort. So there are two types of mistakes that you can make. You can either stop tracing prematurely, so you miss part of the neuron, or you can this is a so-called split error, or you can trace into something else that you shouldn't be tracing into, and that's a so-called merge error, which is actually in practice much worse in terms of the impact that it has. So when we were starting already with those kind of improvements that we have made, we were at an expected run length of about 1 millimeter. So this is this blue neuron on the left. Since then, through various optimizations, we were able to boost it by about 1 order of magnitude, so around 10 millimeters. And visually, the difference between 1 and 10 is something like this. So we are now able to trace much larger and more morphologically complex neurons without making mistakes. People sometimes call this metric a self-driving car metric because of this analogy where, if you have a self-driving car, one thing you want to measure is how far can you drive before a human driver has to take over. So this is actually similar. How far can we trace a neuron before an expert has to take over and fix a mistake? The other method that I wanted to mention today is related to this issue of generalizability and the cost of collecting ground truth data, which is something you've heard a little bit about before. And in particular, one issue that we have is that acquisition of ground truth is very costly and it requires expert knowledge. This is normal for the field, but it's also sample specific. So whenever we get a new sample from one of our collaborators, we basically have to start from scratch. We have to re-annotate it, retrain the models, and so on. It's very tedious. So we thought, okay, there's been a lot of progress in the field of computer vision. In particular, there are those things called cyclograms, which are networks that can kind of translate images visually between two unpaired data sets. So maybe you have photos of mountains in the summer and photos of mountains in the winter, and you can actually train a network that is going to convert between summer images to winter images or vice versa. And the simple insight that we have had is, okay, so you can do this on natural images. Maybe you can do this on brain images, which are actually even more similar to themselves than natural images. And maybe it would actually work even better if we had a way to somehow plug in the segmentation model that we have built, this filtering network, into this whole process. And this is exactly what we did. So this is a kind of illustration of what a cyclogram schematically does. We plugged in the red thing, which is a segmentation model. The rest is kind of off the shelf. This is a standard cyclogram. And we tested it to see whether it works. And indeed it does. This is one example from some public data sets called SNEMI and SEGEM. Those are both samples from mouse visual cortex, if I remember correctly, but taken with two very different techniques and in different labs. And if you take a segmentation model that was trained on SNEMI and you apply it to this SEGEM thing, it doesn't work very well. Like you can see there is a lot of green, and this is not what you want. But if you then train this SEGEM model, as we call it, and you do a translation from SEGEM to this kind of SNEMI style, so to speak, segmentation model actually works very well. In this case, if you measure it quantitatively, you can see that it is basically state of the art, as you would get if you were training on this SEGEM directly. And here, of course, it looks easy because there's a very different kind of contrast statistics of those images. But this is just for illustration purposes. There's actually more going on at the kind of fine level of individual membranes and so on that is important. Turns out that this is actually also useful for other things without kind of special retraining or anything like that. It can be used for image correction. So sometimes in those data sets, you end up with areas that are kind of fuzzy or defocused for whatever reason, and those don't segment very well, but if you then apply this SEGEM, it's possible to automatically correct it, and then the segmentation is improved. And similarly, sometimes we are, because of various limitations of acquisition, sometimes we are missing parts of the data set. So this is kind of scrolling through the z-axis of two small data sets, and you will see that the one on the left kind of blinks. This is where the data was not acquired, basically. And you can see that with the SEGEM approach, we can fill it in, and so that at least a human eye doesn't really see anymore, but there is something missing, and also the segmentation, if you measure the quality, is improved. Okay, so to summarize, I think the main point that I wanted to make is that our field of connectomics is one that demands kind of extreme accuracy and scale at the same time, and it's a complex field, so we benefit a lot from working in a collaborative style. And machine learning is obviously very important. You can get a lot done with off-the-shelf approaches, but to really be at the bleeding edge of what's possible, it's necessary to do customized approaches. And if you're interested in our work, please do check out our website. And we also release quite a lot of open source software. So if you work with large-scale 2D or 3D images, those are interesting things to look at for visualization, storage, and alignment. Thanks so much for your attention.
Thank you very much, Michał, for this very, very interesting talk. Definitely your paper on flood filling networks has jumped to the top of my reading list now, so
Thanks.
definitely check it out. So do we have any questions from the audience? Here you go.
Thank you for your talk. You mentioned that one of the parameters that makes like a collaboration work was that you have a large and complex problem. What are issues that you encounter when you have like small collaborations that make it not work in your case?
Well, I would say maybe not necessarily, like the scale is interesting in the sense that there's overhead in setting up a collaboration that we run into. And, you know, because of the size of the projects, they mostly take, I would say, you know, more than a year usually a few years. So if you look at it from that point of view, the overhead is usually hidden, right? So like okay, the people have to meet, get to know each other, understand what they're all doing. That takes, you know, days to weeks maybe. If it was a small collaboration that only takes 2 months, that's a big fraction of the overall time. If the collaboration takes 3 years, then it's almost irrelevant at that point. So that's one aspect maybe to be aware of.
Thank you.
Thank you. I have a similar question. Why, as maybe naive, why does Google make research on the brain in the first place? Particularly because it's a risky domain. It might be as a risky.
Why is it risky?
Yeah, I mean, how can I say it? In a certain way, what you are saying here, or what you help a lot for looking at the pictures, but let's say maybe it could be that eventually the super breakthrough is done by Harvard. You have provided the pictures, the nice pictures, and then the nice, you know, the big insight comes from somebody else. So,
well, I mean, so I think we would consider this as success if we publish one of those data sets, and now so actually that is in some sense something that we are trying to do, right? Like for those large data sets, we publish them as a resource for the whole community in the hope that somebody will look at it and find more interesting things that we have done. We try to do analysis on them, and sometimes, you know, we find those interesting things, like some of those that I've been showing you. But the hope is that actually the community will use them and find more stuff. And you know, that's great that we do so. Hopefully we'll get citations or something, some sort of credit, but yeah, we don't necessarily need to be the ones who make the core insight from those data. So from that point of view, for us, it's not a risk. It's
okay.
Yeah, would be expected.
Thanks.
Hello, thank you for the interesting talk. So my question is related to the reconstruction error. So like you mentioned in 2016, it was around 1 mm, and right now it's around 10 millimeter. So what is the target goal that you are trying to reach before you think it's sufficiently accurate?
Yeah, that is a very good question. And unfortunately, it is a moving target in the sense that people, in the field, there is always a desire to map larger and larger samples, right? If you think about it, a cubic millimeter is a tiny fraction of a human brain, maybe 1/1,000,000th or so, about 1/500th worth of a mouse brain. So it's really tiny. And of course, the field would like to do a whole brain, right, of a mouse, and maybe at some point a human, probably not in the next decade, but who knows, maybe in a couple decades. And the problem is that, of course, the error rates that are acceptable, they get lower and lower the larger a sample is, because of various reasons. Like mathematically, you know, you at some point you get above a kind of percolation threshold, and then everything is useless if you get too many merge errors. But even if you don't, and if your kind of error density is constant in space, the larger the space, the more errors you have, and the more kind of human effort will be required to fix it. So our goal from a kind of maybe practical point of view is to minimize the human proofreading necessary after we do the reconstruction. So you saw on the Hemi brain, that was 50 years, that's still quite a lot, right? We would ideally be at a level where for a single sample that would be almost negligible. So that, you know, you could imagine completely automating this whole thing where a lab could take a scan, apply the algorithms, and then directly go to analyzing the data without worrying about the errors. And what that translates to in terms of this expected run length, I think nobody really knows at this point. In terms of cable length in the brain, it is many kilometers. So certainly once we get in the like meter or kilometer range, things will be very different. But yeah, I think there's probably a few orders of magnitude left worth of progress there.
So maybe we just take one last question. Here you go, Simon.
Thanks a lot for the talk again. I'm going to make it two short ones, but you can just answer one. So I understand that Google normally sort of asks himself, what is our unfair advantage before they take on a project? So my first question would be, what is your unfair advantage here? And the second one that you can choose from would be, how, if at all, can this be monetized at all, given that you're still a company?
Okay, so I should answer both or one? Maybe both. So the unfair advantage, if you might call it this way, is the compute, right? So one thing that we have access to is a lot of computational power, and indeed, if you think about it, 1.4 petabytes processed by a relatively expensive machine learning model, that requires a lot of compute. So we have access to that, and we actually try to be very kind of economical about how we use it. So we are kind of scavengers within the company to some degree. So we use resources that other people are not using at the time. Turns out there's quite a lot of those, so we try to make good use of them for science in this case, right? In terms of monetization, I don't think that's an angle right now. Maybe in some faraway future, you can imagine connectomics becoming as popular as genomics is these days. So maybe this could be a service that they could provide to researchers. But right now, the scale is not there, and also the automation is also not there at that level. So we are not doing it for the money. We are doing it for the goodwill of the community and the progress that we hope it will generate.
So Michał, maybe a follow-up on that question. So you showed one of the very nice pictures where you had like several of the neurons which were connected. So could you give a rough idea of the compute that was required to fill that network?
For this particular data set, probably on the order of hundreds of thousands of GPU hours, maybe more. I just don't remember the details. Like, it's in our paper. It's actually publicly available. I just don't remember the number off the top of my head, but a lot.
And maybe just one last question. So I don't know much about the connectomics, but at what point can you also say that you can just repeat the patterns? Like, how far do you have to actually really resolve it?
You mean like physically within a brain?
Physically, yes.
Yes, so that depends on the brain structure. For instance, this hemibrain data set, so Drosophila, or actually most of brains in biology, they're spatially symmetrical, right? So the left side and the right-hand side, they're kind of similar. So if you're only after large-scale structure, then maybe you can do half, or sometimes, researchers are very interested in a particular region of the data set. Sometimes from prior studies is known that, for instance, olfactory processing, like early olfactory processing happens in the olfactory bulb. So then you only scan that part and not the rest. But I have to say, there's always, in the back of your mind, there's always this will for more, because even though the brain is compartmentalized to some degree, it's all also very interconnected. So whenever you stop, you're always losing something, and people are always wondering, okay, so what are we losing by not going further in this direction? So if you ask any neuroscientist working in this field, they always want more. I don't think there's like, oh, that's enough. We don't need any, we don't need to go further in this data set. But also, I think maybe as the field kind of matures and there's more and more prior studies available, at some point people will realize like, oh, okay, like everything we need is contained in this region. We don't need more in this case. But we are not there yet. For now, people just want more.
Thank you, Michał. Very interesting. Thanks a lot.
Sure. Thank you.